Okay, so today we are going to be focusing on protein synthesis, which includes both transcription and translation. And to start off, you're going to, again, have to take the sequence 1 molecule and build it. So find the pieces, boom, and there they are. That'll probably take you a little bit longer, but that's okay. And the goal is we want to use the information of this DNA-based sequence and use it to build an actual protein. So somehow we need to translate this sequence of DNA bases into a protein, a sequence of amino acids. So the first step is transcription, and that takes place in the nucleus. And I have over here an enzyme called Rna Polymerase that's going to help us do that. And what happens is this double-stranded DNA molecule is going to feed into this enzyme, and RNA polymerase separates the two strands from one another. Now remember that these strands are held together by hydrogen bonds, which are relatively weak compared to the covalent bonds holding each nucleotide together. And so we can pull these apart fairly easily. And then there's an active site here that fits the next RNA nucleotide. So I'm going to grab the RNA nucleotide. Since this is a G, C is going to be complementary, so it fits in there, and a new hydrogen bond is formed. Now note that the RNA nucleotides have a slightly different sugar group than the DNA nucleotides, and that's why these are square while these are circular. Kind of represent that that is a different type of molecule. So in transcription, what happens is we're just going to make a disposable copy of this DNA molecule. So I have my G, and then I slide over, and then there's another base that goes on there. And then the new RNA strand is actually going to slide out this way. And so as I keep going, the next one is a G, boom, and then a C, boom, and then an A, boom. And you kind of see that as my RNA is being built, the DNA continues to feed through there. And eventually I get to the U and the G. And this process just keeps on going. The U, and eventually, let's kind of stick these all together at once. The G, U, and G continue one at a time. Now you'll notice that on the other side now, the DNA molecule is reforming. And so we're sort of breaking it apart in order to produce this messenger RNA, and then we're reforming it over here on the other side. And so by the time we're done with this, the DNA will be left just how it was, and our messenger RNA will be fully built. And in order to save time for the video, I went ahead and pre-built it, and I have this over here, and I'll just kind of stick that on there. So if we'd carry this out to for the whole length of the DNA molecule, this is the RNA sequence that would have arisen, and it is complementary to this DNA-based sequence. So I've succeeded in making a disposable copy of the genetic information, the sequence of bases, in this RNA molecule. So that is the end of transcription. Now what's going to happen is we're going to take this copy and we're going to bring it out of the nucleus to a ribosome, and we're going to use this to make a protein. That process is called translation. So let's walk over to another lab table to represent that we have a slightly different process happening here. And the RNA molecule that we just made is going to feed into this ribosome. You'll notice there's 3 different sites. There's the A, think amino acid. That's where the new tRNA molecule with the new amino acid is going to come in. P, this is where the growing polypeptide is held. E, that's where the tRNA molecule is going to exit. So we've got our ribosome, we've got our RNA strand, and then we have a series of tRNA molecules. Note that this is the translator between the language of nucleic acid, a sequence of bases, and an amino acid. So this part is called the anticodon, and it's going to interact with the mRNA. And this part is this is the amino acid up here. So every protein starts with a methionine. AUG is the start signal. So I have AUG of my mRNA strand, and so the appropriate tRNA is going to come in. If you look up AUG on the genetic code, we can find it over here, AUG, and it says it's codes for methionine. And sure enough, this tRNA molecule, the anticodon, interacts with the codon perfectly. Boom. And on the other side of this is methionine, our appropriate amino acid. So AUG codes for methionine, and tRNA is what actually positions it. So that's going to go in there. This is then going to slide over. It's now being held in the P site, which exposes a new 3-base sequence or codon to the A site. UGU, if we go to our genetic code, UGU codes for cysteine. And sure enough, the next tRNA, the anticodon, ACA, is complementary to that. And it has the amino acid cysteine. So our tRNA is helping me translate these 3 bases, the codons, to an appropriate amino acid. And then what happens is a new peptide bond is formed. This methionine is going to attach to the cysteine. This is going to slide over. Note that the growing polypeptide is now in the P site. The now empty tRNA is in the E site where it's going to exit. That's gone, or it can be recycled later. And the A site is now available for a new one. So our next one, GAG, if we go to our genetic code, GAG codes for glutamic acid. And lo and behold, here's our anticodon CUC, which interacts perfectly with that. It's got glutamic acid. We transfer, ba-boom. We slide. This one goes away. And we just keep repeating this process. So the next one is going to come in, AUA, which codes for isoleucine. That's going to attach. It's going to slide. The next one, CAU, is going to code for histidine. It goes in. It attaches. It slides. The next one, UGG, codes for tryptophan. It attaches. It slides. CCA codes for proline. It attaches. It slides. And you can see how this works, and I'm continuing for a reason. If you're getting bored, hang in there with me. A couple more here. The next one is histidine. And finally, cysteine. Oops. Now this next one is interesting. So it goes in, attaches, and slides. The next codon is AGC. And if you, whoops, no it's not, it's UAG, it's UAG. And if you look over here, UAG says stop. So that's the stop codon. And so what happens is, this protein comes in and attaches to the stop codon. And now when we try to transfer this protein on, there's nothing for it to attach to. And as a result, it just kind of falls off, and that protein is now part of the cell and is free to do its job. It's going to fold spontaneously as this is happening and then ultimately be used to do its job. So notice that by the time we're done, and these should all have exited, I forgot to take these off, so we'll just yank those all off. So notice that through this process, we were able to take the information in DNA. We made a disposable copy through the process of transcription, which we call mRNA. We use that disposable copy to put a very specific sequence of amino acids in order, which becomes our protein. So the DNA codes for protein.